Alright guys, welcome back to Underground Science, and in this video we'll be talking about DNA replication. First things first, we should know that DNA replication occurs in a semi-conservative manner. That means that if we start with these two strands, right, in blue is going to be the old parent strand, right, and then orange is going to be the new strand that we replicated, that occurred as a result of replication, that's complement to the old strand. So we can see the top 3' to 5' strand, we take that, replicate it, and get a new DNA, new strand that's perfectly complement to the old DNA, which is in blue. And then we take the other old blue DNA strand, the parent strand, right, 5' to 3' one, and we replicate that and get a new orange DNA strand. So we can see we conserved the old parent DNA while making new DNA that's complement to the parent or old DNA. So that's semi-conservative. That's why DNA replication is semi-conservative, because you conserve the old parent DNA, which is in blue, but you also, in the process, make some new DNA, which we're showing in orange. Alright, so first things first, we form an origin of replication. There's multiple origins of replications in eukaryotes, but in this case, right now, what we're showing is first, right, in order for DNA replication to even start, you need your bases or your nucleotides to unwind. So you need an origin of replication, a place where replication can begin, okay? Because once you unwind the DNA, like shown here, right, in this photo, this origin of replication, then only enzymes like helicase can come in, right? So helicase can come in and start unwinding DNA that way or unwinding DNA that way. All right, so let's go and see how that works. So this is helicase in our picture, right? Helicase right there in red. And it's unwinding the DNA that way. So DNA replication, right? DNA replication is occurring that way. All right? So helicase is unwinding the DNA. And basically what it does is so if we write that down up here, right? Helicase unwinds the DNA. And on top of that, what we should know is that helicase uses a lot of ATP. Okay, so helicase needs a lot of ATP in order to do its job. And the way it unwinds the DNA is, remember, what's holding the nitrogenous bases together, like, for example, adenine binds to thymine with two hydrogen bonds, while cytosine binds to guanine with 3 hydrogen bonds. So if helicase is unwinding the DNA, that means it's breaking the hydrogen bonds. Okay, so that's what it's doing. It's breaking the hydrogen bonds between the nitrogenous bases. Okay, so I'm just going to say breaking H bonds or hydrogen bonds. All right, so it's unwinding the DNA that way. So DNA replication is occurring that way, right, following helicase. And, well, if it's unwinding the two strands, right, so the top one is going to be the 3' to 5', the bottom one's going to be 5' to 3'. So if we should just name it right now, the top one, because the top one is getting synthesized as the helicase is going in that direction, this is the leading strand, okay? This is the leading strand. The top strand is the leading strand. And the bottom strand is going to be called the lagging strand, and we'll see why in a moment. All right? So the bottom strand is called the lagging strand in this case. So what we should know first is, right, the first stage of DNA replication is called initiation. So that's when your origin of replication forms and helicase comes in and starts unwinding the DNA. Okay, another thing we should know about initiation is what's keeping these strands of DNA apart? Why can't they just collapse back together and reanneal, right? So the answer to that are these single-stranded binding proteins on each strand. That holds these two strands apart, okay, so they don't reanneal and come back together, okay? And another thing we should know right now, because we're on the topic of helicase and unwinding the DNA, all right, is that while helicase is unwinding the DNA, ahead of it, we form supercoils. Basically, the DNA ahead of it starts tying too much, okay? Starts tying up too much. Those are supercoils. We have an enzyme to prevent that from happening. That enzyme is called topoisomerase, okay? So topoisomerase is ahead of the helicase and making sure that we don't form too many supercoils, okay? Which is going to be important when we talk about certain drugs targeting topoisomerase. All right, so that's initiation. That's basically it. When we form an origin of replication, okay, so an area where DNA replication can start, so right here, And then helicase is involved in initiation, where it starts unwinding the DNA. We have single-stranded binding proteins involved in initiation, holding the two strands apart, the leading and lagging strands apart, so they don't re-anneal back together. And we have topoisomerase helping us to basically, it's helping us to not form as many supercoils ahead of helicase, right? Because if you form too many supercoils ahead of helicase can't do its job properly, all right? So that's initiation. Now we start elongation, where we actually start replicating the DNA. So first things first is, let's focus on our leading strand for now, okay? First things first, we need something called a primer, which is going to be RNA, okay? So I know it's weird because it's DNA replication. Why do we have RNA? But we'll see later, all right? This is actually an important step. So let's say first we need a primer, okay? Now who's gonna provide us that primer? That enzyme that's going to provide us with the primer is called primase, okay? So this is going to help us, or this is going to provide us with the RNA primer. All right? And so one thing we should know is that in DNA replication, usually what's happening is we're forming our nucleotides from the 5' direction to the 3' direction. Here's what that means. So we see our leading strand, right? It's 3' to 5'. So I just said in DNA replication, what we're doing is we're forming our nucleotides from the 5' to 3' direction. So if this is the 3' to 5' direction, we're forming our nucleotides from the 5' to 3' direction, which means this side, if we're forming a new strand, right, replicating new strand, so let's say we put down our primer, right? If we put down our primer right here, let's say it's only 2 nucleotides long, and it's RNA, okay? So the complement base to adenine in RNA is uracil, and then thymine is adenine. So we have a U and an A, that's part of our primer. Notice we started the primer here and synthesized it that way, okay? We're synthesizing the primer that way. Why that way? Because this is going to be from 5' to 3', okay? Remember 3' is where our OH group is, okay? So we're synthesizing from 5' to 3' primer. Now, remember, you need the 3' OH group in order to add nucleotides onto, okay? So next, what's going to happen is DNA polymerase 3 will come into the picture, okay? So DNA polymerase 3 will come in, which is in prokaryotes, and it will add on to the primer, okay? So DNA polymerase 3 will come in and add on to the primer now, onto the 3' end. So let's go and erase this, and let's see what happens. So we get our bases now adding on from the primer. So we're going from 5' to 3', because what DNA polymerase 3 does is, so DNA polymerase 3 will read, he or she will read the strand from 3' to 5', okay? The DNA polymerase 3 will read the strand from 3' to 5', okay? It's reading it. So it's like, okay, let's start here, right? Because we formed the primer already. So it's reading it from 3' to 5'. So it's saying, okay, this is C, G, C, all right? So I'm reading from left to right, which is 3' to 5', but it's synthesizing from 5' to 3'. Okay, so this is really important. So we're synthesizing, though, from 5' to 3'. Synthesizing the nucleotides from, right, synthesizing from 5' to 3', okay? So again, it's reading from 3' to 5', so it's reading this way. DNA polymerase is reading this way, DNA polymerase 3. So it's reading from C, okay, the next is C, G, all right, cool. But then it's synthesizing from 5' to 3', okay? Because remember, DNA is anti-parallel. So if you're reading from 3' to 5', and then you start laying down the nucleotides that's complement to the 3' to 5', then you're synthesizing it in the anti-parallel fashion. So you're starting at 5' and then synthesizing it from 5' to 3'. Okay, so it's going to read 3' to 5', and then we're going to start synthesizing now from 5' to 3'. So now we're going to be like, all right, this is C, right? On the parent strand, we have C. So then the complement to cytosine is guanine. So we're going to say guanine, and then Let's actually go and draw all this out, right? All the individual bases. All right, now let's go write it in. So then there's going to be guanine, then cytosine, then guanine, then not cytosine, then adenine, thymine, adenine, guanine, cytosine, right? Those are all the complement bases. So see what we did. So here, let's erase this 3', okay? So we just synthesized the new strand from 5', okay? From 5', let's go and fix that, sorry. We synthesize it from 5' to 3'. So this is now 3'. Okay, cool. So that was DNA polymerase 3 on the leading strand. Notice how on the leading strand, we only used one primer, right? You didn't see multiple primers getting added. Why is that? So why is this called a leading strand? Because we're literally synthesizing the new strand in the same direction as helicase, okay? So we're going with the replication fork, with the direction of helicase. Okay, but look what happened to the lagging strand. Look, so the lagging strand, right? What we have to focus on is that this starts at the 5' end, and then it ends at the 3' end. So, but remember what we said DNA polymerase 3 does. Okay, DNA polymerase reads from 3 to 5. So 3 to 5 is this way in the lagging strand, and it's going to synthesize from 5 to 3, which is this way. Because remember, when you're synthesizing the opposite of what you read, okay? So if it's reading 3' to 5' this way, it's going to synthesize the same way but opposite because it's anti-parallel. It's going to synthesize from 5 to 3. So that means we're synthesizing new nucleotides this way, okay, to the left. So the lagging strand is going in the opposite direction of the replication fork in helicase. Okay, so the lagging strand's going this way, right? But the direction of DNA replication is going that way. So that's why it's called a lagging strand, okay? So what happens is, again, a primer is going to come in with the help of primase. So let's say we put a primer right here, right? Let's say this primer is also 2 nucleotides long. So first, as we know, that this strand, right, this 3 to 5' strand is going to be complement to this 5 to 3. So let's go and feed that in real quick. So this is going to be thymine, adenine, guanine, cytosine, guanine, adenine, thymine, adenine, guanine, cytosine, right? We put a primer. Again, when you're putting a primer, we're putting that from 5 to 3 direction. So that means this is the 5', this is the 3', where the OH is. Okay, so we put the primer in the 5 to 3 direction. So let's write the nucleotides in the primer. So remember, primer's RNA, but in this case it doesn't really matter because remember RNA doesn't have thymine, it has uracil instead. So that's cytosine. All right, cool. So remember, after we put the primer, we synthesize the DNA, we replicate the DNA. Which direction are we replicating DNA in? Remember, DNA polymerase 3 will now come and read from 3' to 5', which is this way, okay, this way. Because see, this is 3', this is 5'. DNA polymerase 3 will read from 3 to 5 this way, but it will synthesize from 5 to 3. Remember, when you synthesize nucleotides, DNA polymerase needs the 3' OH end. So it's going to start here, okay, so we're going to synthesize that way. Okay, so let's go and draw that in. So we're going to synthesize from starting at the 3' end of the primer, okay, because we're synthesizing now from what? This is the 5' end, and we synthesize from 5' to 3', okay? So what did we just make? We made thymine and adenine, right, because it's DNA. Now, imagine this, so you just put the primer here and we synthesize DNA this way. But this helicase is not stopping, it's going that way. So imagine we're getting more and more bases on this side. So if we imagine, then another primase will come in and put another primer. Let's say it puts another primer over here, right? And then this is going to be U and A, right? Again, in the 5 to 3 prime direction, right? 5 to 3 prime direction. Then DNA polymerase 3 will come in. On the 3 prime end, it'll start adding, right? It's DNA nucleotides. So what will that be? So we're going to have here, here, and here, right? So that's going to be thymine, cytosine, guanine. Okay? Again, primer is going to come in. A primase is going to come in, lay down a primer. Let's say it lays it down right here. Okay? Then we're going to have, what are those bases going to be? That's going to be guanine and cytosine. Then, because it laid it down in the 5 to 3 prime direction, this is the 3 prime end of the primer. Okay? So we're going to have DNA polymerase 3 coming here and laying down its nucleotides this way, left. Okay, this is 5 to 3 direction. So we're going to have that again, right? It's kind of repetitive, but we'll see why at the end. Boom, boom. Okay, so what's going to be inside there? Well, DNA polymerase 3 will put the complement to the bases. So complement of guanine is cytosine, complement adenine is thymine. So next what happens is, like, look, notice what we have so far. Look at we've been using multiple primers on the lagging strand. Okay, so this alternating sequences of, okay, we have a primer, then DNA, then again primer here, then DNA, primer, DNA synthesis, right? This alternating sequences of DNA primer, that's called a term which we should know, which are Okazaki fragments. Okay, because the lagging strand is synthesized in fragments. Okay, because we have primer, synthesized DNA. Then you go more ahead, primer, DNA. These are called Okazaki fragments. Okay, so let's just write that down. Acknowledge the fact that when we're synthesizing DNA in fragments in the lagging strand, those fragments specifically are called Okazaki fragments. Okay? This alternating sequences of primers and DNA, multiple primers and DNA on the lagging strand. Okay, sweet. So the next question in elongation is, well, this is DNA replication, so we can't have RNA here. So who's going to remove these RNA primers and replace it with DNA, right? So that next enzyme we have to talk about is DNA polymerase 1. Okay, so next enzyme we have to talk about is DNA polymerase 1. Okay, which again is in prokaryotes. DNA polymerase 1, what it's going to do is, right, all we have to do is talk about it. So it's going to replace the RNA primers and then replace it with DNA, okay? So the RNA primers are from, remember, how do we synthesize the primers? We went from 5 to 3 prime, right? 5 prime to 3 prime. So that's why DNA polymerase 1 is going to have something called 5 prime to 3 prime exonuclease activity. This is responsible for taking out the primers, okay? Taking out the primers. Okay, so DNA polymerase 1 will take out the RNA primers via its 5 prime to 3 prime exonuclease activity, okay? So again, these RNA primers, remember, we synthesize them from 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime, right? So it's going to take these RNA primers out and replace it with DNA, complement, like whatever the complement bases are, right, to this strand, if we're talking about the lagging strand. Okay, so it's going to replace it with DNA. Additionally, DNA polymerase 1 also has something else called 3 prime to 5 prime exonuclease activity. It's specifically for proofreading. Okay, so 3 prime to 5 prime exonuclease activity for DNA polymerase 1, which is basically for like proofreading, for example. And also, what we should know is that DNA polymerase 3 also has 3 prime to 5 prime exonuclease activity for proofreading. Okay, that's important to note. So like, for example, if DNA polymerase is laying down its nucleotides, right, from 5 to 3 prime, and then it senses like, okay, wait, I put a wrong nucleotide in, right, that's not complement, then it can use its 3 prime to 5 prime exonuclease activity in order to proofread and put the correct nucleotide in. All right, so let's say we're on DNA polymerase 1, right? So what did DNA polymerase 1 do? It takes out the primers, okay, and it's going to replace that with the correct complement DNA, okay, to the parent strand. So let's say we take these primers out. It's going to get a little messy in here, my bad. But hopefully, we understand the basic concept of DNA replication. So let's put this here. And then we're going to have the DNA polymerase 1 will take out the primers and put the correct DNA nucleotides in, which is going to be, in this case, we're going to have C and C here, and then T, A, and then C, G, right? And then let's encircle all that with our colors. Okay? But what happens is we're going to have certain spaces here, okay? So this won't be fully connected together, okay? Because what happens is we need DNA ligase to fully seal the nucleotides together. Okay, so if we're talking about like this one, and then let's say we formed the space right here in between these two, right? Let's just take these two examples right here and right here. Let's say we replace the primer, right, with the correct nucleotides, and then we need to form proper seals because this isn't fully sealed together. So if we look at this space right here and this space where the primer was, right, and then this space, like we need to seal these spaces. So what the last step is, after DNA polymerase 1 takes out the primers, puts in the correct nucleotides, DNA nucleotides, then we seal these spaces with DNA ligase. Okay, so it just seals the phosphodiester bonds together. So then lastly, we use DNA ligase in order to seal the phosphodiester bonds together. Okay, so DNA ligase. So that's basically elongation. It's like first you put primers, then you use DNA polymerase 3 to synthesize your strand. Then you take out the primers, right? And we also take out this primer, by the way. I just didn't show it because we were trying to focus on the lagging strand. But if I did show this, you would take out this primer, right? You take out this primer, and then what you would do is you would put, the necessary DNA nucleotides on it, especially if we had like more we didn't show all the nucleotides in this video, because there's this DNA strand could be way longer, but we're just trying to focus on how this process of taking out the primers works. So first you take out the primers with DNA polymerase 1, right? And then DNA polymerase 1 also puts in the correct DNA nucleotides also. Okay, so that's important to know. We could have showed more nucleotides here, right, on this side to make it more clear. But it's fine. We get the concept, right? So DNA polymerase 1 will take out this primer, and then we'll put the correct DNA nucleotides in with the help of DNA polymerase 1, and then we seal the phosphodiester bonds with DNA ligase. So if we take this primer out, right, notice that this is synthesizing from what? What is this? 5' over here to 3'. Remember, DNA polymerase needs the 3' end in order to make nucleotides, okay? So we still have that 3' end here, so DNA polymerase can come in and put the correct DNA nucleotides in, which in this case would be, let's change the color, A and T, right? That's basically it. And then remember, since we had the primer here, we're going to have a space right here, not fully sealed. Then DNA ligase will come in, and then it will seal the phosphodiester bonds together. All right. So that's elongation, and then termination is basically, if you just imagine like another helicase on this side, because remember, we can also have something like this where you have like another, because remember, eukaryotes have what? Multiple origin of replications. So you can also have another helicase, right, working like this side, going this way, and then these helicases can meet together, and then you just basically terminate DNA replication. All right, so that's basically it. So yeah, that was DNA replication. Probably too long of a video, but hopefully you guys understood DNA replication, and then we'll talk about certain drugs targeting certain enzymes and maybe certain diseases too in DNA replication in our next video, and then yeah, go from there. Hopefully you guys enjoy this video, and thanks for watching.